Today we explore the groundbreaking work of Dr. Geeta Narlikar from UCSF. Her research unravels the molecular mechanisms that shape our genome, revealing how chromatin remodelers influence everything from cell fate to disease progression. Stay with us as we delve into the molecular machines at the heart of life's complexity.
Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello, Geeta. Welcome on the show.
Thank you for inviting me to this podcast.
Yeah, I'm very happy to do so. So currently you are a professor at UCSF. Maybe we can start with the question how you became interested in biology in the first place and then in pursuing a career in science.
That's a great question. In undergrad, I was initially very interested in physics. But then I got introduced to chemistry and found my way to biology through chemistry. And so it became very clear in my undergrad that a lot of biological questions could be addressed through thinking through the concept of chemistry combined with physics. And so I entered biology through physics and chemistry. But biology was where, to me, the biggest questions were, the most exciting questions were, and that's how I got into biology.
So when looking at your PubMed record and comparing the first author papers then to the last author papers, there is a clear shift in things you have been doing. So how did you then end up after your studies in the field of epigenetics?
How did I land up in the field of chromatin itself? Is that the question?
I mean, that's to be debated, right? Is chromatin epigenetics or is it not?
Yes. So my graduate work was on catalytic RNA.
Okay, yeah.
And I learned how to think like an enzymologist in my graduate work. And after my graduate work, I wanted to take on a more complicated problem than catalytic RNA. And around that time, chromatin remodeling machines were being discovered. And so that drew me into the chromatin remodeling world in Bob Kingston's lab. But so once I was in Bob Kingston's lab, I was hooked on chromatin. Right? And it's through working on chromatin remodeling machines that I realized there was a huge playground of questions that I could address. And so you could say that from the time I entered Bob's lab, I was in the chromatin world. And the epigenetic world is just, added on top of chromatin, right? So it's not distinct in my mind.
Yeah, okay. So how did you then end up at UCSF?
So after my postdoc in Bob's lab, I was interested in an academic career. I really enjoyed biological mysteries, solving biological problems. And so the academic career seemed right for me. And I applied, as many postdocs do, to academic jobs across the country. And UCSF was the one place where I felt I would get excellent students. And that has borne out to be true. Most of our lab's creative discoveries have come through students. And I really enjoyed interacting with the colleagues. So it was a combination of students and colleagues that drew me to UCSF. And I've been here now for 21 years almost.
That's great. So let's come to your science that centers around chromatin remodeling and HP1 and phase separation in heterochromatin. I want to start with a paper that was very interesting, that has a very interesting title, which is Distinct Strategies to Make Nucleosomal DNA Accessible, which seems to be at the core of everything, basically. You need to make things accessible, right? There you compared two prominent human ATP-dependent remodelers, BRG1 and SNF2H, for their abilities to make a spectrum of nucleosomal sites accessible. Can you talk about this study and what you found?
Yeah, so this is an interesting study. This was my last paper from Bob Kingston's lab. And so Bob decided that I should be the corresponding author on it. And, Bob has been an amazing mentor throughout, and he still is. The first author is a really good friend, Wang Fan. And the two of us worked on the story together. And the context for this story is around that time, there was a view in the field that different chromatin remodelers, so SNF2H is one, BRG1 is the other, worked by the same mechanism. Okay? And this is a little bit of a philosophical distinction, right? What do you call mechanism? And I can say where we are now, full circle, whatever, 25 years later. But then there was this debate in the field whether SNF2H and BRG1 type enzymes are essentially doing the same thing to nucleosomes. Okay? And they're not that different from each other. So we decided to tackle this kind of through the biochemical lens and say, hey, let's compare the two side by side in multiple assays. And what we found is that each of the two enzymes were causing different types of products. They were catalyzing the production of different kinds of products. Okay? So they were clearly different, right? And in the discussion, I'm pretty sure we wrote about how their core mechanisms might be very similar. So the folks in the field who are saying they're similar are not wrong, but they're both using their core mechanisms to generate different outputs. It's the same machine, presumably acting in the same way, but then the ATPase actions are coupled in different ways to generate different products.
Can you maybe
Can
you maybe say what the different products are, just to make it complete?
So the different products that we found then, right? And again, this was 2003, was it? The paper, if I'm remembering right? It came out in 2003. It was a while ago. What we found was that BRG1 could create a diverse range of products. It could create nucleosomes that were slid off the ends of DNA. It could create nucleosomes with altered conformations. But SNF2H was much more calm, right? It's a calmer machine. It would take a nucleosome, move it from the end to the center, and nothing else. And so it was quite interesting because the two are implicated in different biological roles. And so it was very satisfying for us to see biochemically, for Wang and me to see that the products reflect the different biological roles assigned to these two enzymes. Does that help?
Yeah. And what did you find about the different? I mean, if you have BRG1 that clearly might, how would you say that? Like, not destroy, but kind of destroy the nucleosome then, and the SNF2H just move it around and say, well, let's make it accessible and then come back later. So, yeah.
It's interesting to assign personalities to remodeling complexes. But and the thing to remember and the thing to really kind of be aware of is science is step by step, right? So what we learned then was what the core ATPase subunits were capable of doing. We know that these core ATPase subunits are part of larger complexes. And so then what we left open was how do these other subunits modulate the activity and the outcome of the core ATPase subunits, right? And so now, if we come fast forward to the current day, we know a lot more, right? We know that the other subunits of BRG1 might alter its output, might direct it into a few different types of output. Whereas the other subunits of SNF2H still make it just move nucleosomes back and forth, but might alter the directionality in which it moves. But I think from the perspective of mechanism, it was important for Wang and me to say, what are the core ATPases capable of? And yes, BRG1 is more disruptive, consistent with its role in transcription activation. You want that region of the genome to be accessible and available to transcription factors for elongating polymerase and all of that. SNF2H, when it's part of the ACF complex, is important in generating heterochromatin. So you want things to be evenly spaced. That is the model. And that even spacing of nucleosomes might then allow folding and compaction of chromatin.
So what you then did is that you focused more on the ACF, ATP-dependent chromatin assembly factor. And this, as you already hinted, generates regularly spaced nucleosome arrays in vitro and is required for transcriptional silencing in vivo. As you just said, a key unresolved question is how ACF measures equal spacing between those nucleosomes. So how did you tackle that problem and what did you find there?
So that was a really fun study. So this was like the first major study from my new lab at that point. And there were two amazing folks in the lab, Tina Shayan, who was the first technician to join the lab, and Janet Yang, one of the first students to join the lab. And the two of them teamed up for this study. So it's an interesting problem, right? You give this machine that uses ATP nucleosomes, and it can space them. It somehow has a capacity to measure the distance between nucleosomes. So how do you even conceptualize a problem like that? And there are many different models you can imagine would explain that, right? You can imagine a model where ACF has a ruler. It has a subunit or part of a subunit that measures and binds to the DNA coming out from a nucleosome and says, hey, if it's long enough, I will move it Otherwise, I'm going to just stop, right? And so it could be like a static model. When I go to the right spacing, I stop. Okay. So you move, and you stop. Okay. So that was one model. And that was kind of a little bit of the prevalent model in the field then. And we came in and we said, let's just measure the kinetics of the process, right? So a lot of times you learn a lot about the enzyme by keeping an open mind, right? And saying, I'm going to go in and I'm just going to measure the rate constants for sliding nucleosomes to different flanking DNAs, different lengths of DNA. And let's see what the data tells us. Okay. And so what we found when we did those measurements, and we had to adopt a FRET-based assay to measure those rate constants, those kinetics, because it was more sensitive. It could measure fast kinetics. What we found was that as you made the length of the DNA longer, the enzyme moved the nucleosomes faster. Okay, up to a certain point, and then it leveled off. Okay? But it never really completely stopped. Even if you gave it a short piece of DNA hanging out from the nucleosome, it would still move the nucleosomes. It just much more slowly. Okay? So that gave us a model where this complex was measuring, in quotes, the DNA by a kinetic mechanism, not by a static ruler mechanism. But it would move the nucleosome back and forth, right? And so if it had a choice to move the nucleosome in the longer direction, it would move it faster in the longer direction than the shorter direction. So if you did a single molecule experiment, which we did later in collaboration with Xiaowei Zhang's lab, and you looked at what a single nucleosome was doing, even when it went to the center, which represents kind of an evenly spaced nucleosome spacing, it was moving.
Mhm.
It was moving back and forth, but it was spending more time in the center.
Because of stochastics.
Not stochastics. It was because of the kinetics. The enzyme's activity was faster. So if it moved off center, it would be moved back to the center much faster than it was moved off center. Right. And so that's the mechanism that, for me personally, and hope presumably for the folks working on the study, was very satisfying because it showed the power of quantitative entomological analysis to get at mechanism.
And this also was right in your ballpark, right? When you started with physics and then seeing all those dynamics and things like that. So this must have been very close to you.
Yeah, and it was great because my graduate advisor was Dan Hirschlag, we had lots of kind of fun conversations about kinetics in the context of the ribozyme. But when I told him this story, it was a really fun conversation to go back and chat with him and say, hey, I'm using everything I learned in my PhD for this, right? It's very satisfying.
Even though it doesn't look like it, but it's really true physics. So you then kind of switched gears a little bit and moved away from ACF and focused on Heterochromatin Protein 1, HP1. This recognizes H3 canine trimethylation on chromatin and hence facilitates gene silencing, obviously. You then investigated how HP1 proteins assemble on methylated nucleosomal templates and how the HP1 nucleosome complex functions. I mean, there must be a lot of things going on, right? If, many things needs to be recruited to then create heterochromatin. Can you talk about the study and what you found?
Yes. So I'm going to tell you, if it's okay, a little bit of the backstory of how I started working on this project. It's a very UCSF story. You asked me why I came here. Part of the reason I came to UCSF is I heard that this place has a highly collaborative spirit, and your science can change in positive ways because of your colleagues. Okay? The Heterochromatin project is an example of that. I was not working on heterochromatin. That was not even on my radar when I joined UCSF. Okay? But somewhere in 2007, I think, two other faculty and I, Hitan Malhani and Barbara Panning and I, did a mini course for students. And we said, let's choose a topic where we read papers, and it's a 2-week intense mini course. We read papers in the area that we all want to learn about. And the area we chose was heterochromatin. And a lot of really excellent work was coming out in the POMB system. Okay? And so we said, let's read those papers and let's learn together. It was an amazing experience. So at the end of those 2 weeks, each one of us, Barbara, Hitan, and I, came out with new questions for our own research. One of the questions that was very obvious to me after we read the papers together was there was very little rigorous biochemistry done in this area. And not because, people are not working on it. It was just a lot of focus was on the biological in vivo genetics, right? And there were a lot of interesting questions that were waiting to be answered by rigorous biochemistry. And so that's how I decided that we would start on the Heterochromatin project. And at that time, I had an amazing postdoc in the lab, Basim Al-Sadi, who's now faculty at UCSF. He said, sounds good. Let me take a go at it, right? He then teamed up with a graduate student, Daniele Kanzia, who's also faculty at UCSF now. And that's how the project got started. And I talk about the origins of the project because to me, it's the people that really matter, right? How do you change? How do you go in new directions? It's the people that take you in new directions. And so they got together and that got us started. And that's how we learned how SOI6, the POMB HP1 protein, oligomerizes. When it sits on a nucleosome, it sits in a way where it has a two dimers sitting on a nucleosome with sticky ends. And the sticky ends gave a biochemical and structural framework for how heterochromatin spreads. If you have sticky ends, the two sticky ends would form interactions with each other across nucleosomes and spread. We also found that the spacing between nucleosomes mattered, going back to the ACF study. If the nucleosomes were close enough, then you could better oligomerize and condense the chromatin. So that's how we got started on heterochromatin with the POMB system.
Mm-hmm.
But later, of course, we went into the human system.
One thing that now is very well known and HP1 can do is like form kind of phase separated droplets, right? So everybody talks about like this phase separation now. And you were also working on that back in the days. So can you talk about what this means and how this affects the formation of heterochromatin?
Yeah. So there were two papers, paper from our lab and paper from Gary Karpen's lab, that showed for the first time that a human and Drosophila HP1 proteins could phase separate. And it was a pretty unusual finding. And so it was actually nice that there was another lab who found something similar, because when you find something that looks weird, a little bit unusual, it's nice when somebody else can find it in another system. And so Gary Karpen and I coordinated our stories. And it was another beautiful example of how you can do really great science by coordination. They independently found it, we independently found it, but it was a fun time. So what does it mean? I'll talk about that in a minute, but I am going to focus on the people here for a minute, if you let me. The finding was made by a graduate student, Adam Larson. His project was to use the human HP1 system, HP1 alpha, and it has this beta and gamma as well, and build on what we learned from the POMB system and say, hey, what's different in the human system? Okay? He's an excellent biochemist. And he would purify HP1 alpha, and he wanted to look at the effect of phosphorylation on it, because there was a lot of interesting work saying phosphorylation of the N-terminal tail of HP1 alpha is important for heterochromatin. He would make the phosphorylated protein in bacteria, purify it, would be super clean. But he would keep getting cloudiness in the tube. Okay? Now, if you're a biochemist, seeing your protein turn cloudy is usually bad news. Okay? And so he would show me this, and I would be cloudy, or it would be like glassy, and I would say, hey, spin it down and take the supernatant. That's where the good stuff is. He fortunately didn't listen to me. He was not collaborating, but he was discussing papers in the phase separation area in other parts of biology with his colleagues, with his fellow students across the hall in David Agar's lab. And so he was very aware of phase separation in biology, right? And because he had been up to date with the paper. So he went and he took this tube and he went to my colleague, Roger Cook's lab, put it on the microscope. And he saw droplets. Okay. That's how this started for us. And once he saw droplets and he kind of came back and said, look, this is what I'm seeing. It was in a lab retreat that he made this, he reported this to the whole lab. We got super excited. And then it became a question of, okay, is this an artifact? Is this real? Is there biological meaning to this? Right? And at that point, we could then put all of our rigorous biochemistry and biophysics into play. And you can say, okay, what do we need to do to make sure that we are not being fooled? Maybe we're wrong, right? Maybe this is just an artifact. And that took Adam on a serious journey by saying, hey, what is the structural basis for this? Can it include chromatin? What's the role of phosphorylation? And that led to the paper that came out in 2017. That's the backstory. And to me, backstories are always very, very important because it tells you how discoveries are made. So what does it mean? You asked, what does it mean for biology? I think it's still an open question. One very obvious consequence of phase separation is you organize chromatin and you condense it. And a later paper from our lab using the POMBI system showed us that. Work by former postdoc Srina Sanuli showed that HP1 can bind on chromatin and in a methylation specifically condense it into droplets. And so there's a very obvious way it has an effect. Yeah.
So these are the droplets that you can also see in a light microscope, or do you need to do some sort of staining to make it visible?
No. These are so you see them by without any fluorescence, just bright field. But then if you can use fluorescence, you can do much more quantitative kinds of measurements, right? You can say, what is the density of chromatin inside? What is the density of HP1? Things like that.
Yeah. So is it really like a black or white thing that if you do the immunofluorescence staining, that HP1 would only be in the droplets and then everywhere else it will be rather lowly present?
So let me clarify something here. So these are in vitro reconstituted droplets, right?
Okay. Mm-hmm.
They're very dynamic. So we can't really immunostain like you'd immunostain a cell because when you fix it, the droplets will just collapse. There is no fixation as such in this context. But because it's in vitro biochemistry, you can really label a lot of different proteins and ask how much of this. So to your question, you can say what fraction of the labeled HP1 is inside the droplets and what is outside. Right. And it is hugely concentrated in the droplets. Same with chromatin. The chromatin is almost completely in the droplets. Very little is in the so-called dilute phase.
So is this that also reflected in those genomics like analysis that people were doing, like the TARTs and like all these 3D structures of chromatin? Is this then also like if you do like the orthogonal analysis, is this reflected in those structures?
So that's a really good question. People have speculated that what we and others see in a test tube with chromatin and HP1 those kinds of interactions could contribute to organization of things like TADs. And what Gary Carpen's lab saw was actually in live Drosophila embryos, they saw these heterochromatin puncta, right? And so there, again, it's dynamics that they were following. They could see coalescence of these heterochromatin puncta. So in terms of what could be happening in a cell, heterochromatin often is organized into these regions called chromocenters. And these chromocenters could have the same properties that we see in a test tube. But I hesitate to say it's exactly the same as you see in a test tube because there's a lot of other factors in the cell that might modulate the behavior. But a lot of these heterochromatin puncta have been shown to be dynamic, have been shown to be consistent with what you would expect for a phase-separated state. But to answer your question from the most biophysical and biochemical perspective, the interactions that we are seeing in a test tube are going to exist in a cell, right? We are looking at interactions that are formed from conserved regions of the protein with conserved regions of histones. So those interactions are going to exist in the cell. Whether they manifest as macroscopic droplets or whether they manifest as a small assembly is going to depend on the context.
Okay. So another protein that you looked at is AT. So maybe we can also talk about how this AT protein can be seen in the context of the other proteins that we talked about in the beginning, like the BRG1 and the SNF2H. Yeah. And how AT then catalyzes nucleosome movement, nucleosome sliding.
So there are about maybe 2 papers that came out in the last 5 years on NO80 from our lab, and I'll talk about 3. There are 3 stories. In the very first story on NO80, and I'll try to summarize it in briefly as much as possible. The first story is from a former graduate student, Coral Zoe. She wanted to study a complex nobody else in the lab was studying. That's how, again, the student driving it, that's how we landed up working on NOAD. She said, I want to figure out something new. I'm going to take on a new challenge. She brought NOAD to the lab. And it's her work in collaboration with a postdoc, Stephanie, who did some really beautiful single molecule work where we learned that NOAD also senses the length of flanking DNA, like ACF, but through a very different set of kinetics. It doesn't make the nucleosome go back and forth very quickly. It latches onto a nucleosome, and once it decides it's going to go, it is very processive. It'll move the nucleosome in one direction. And once in a while, it'll turn around. Right? But NOAD is found to play a role in multiple kind of areas of genome regulation, transcription, DNA repair, replication. And so what that got us interested in is to understand what is NOAD doing and how is it doing it to nucleosomes? What is it exactly doing to nucleosomes? So the first paper told us it is super sensitive to the length of the DNA, even more so than ACF. Okay. It shows a switch-like behavior. What we are now finding, and I can tell you this is unpublished data, but I've been talking about it, so I can talk about it here in this podcast, is that the length sensing of NOAD is determined by an unusual auto-inhibition mechanism. And this is the work of amazing graduate student Upneet Kaur, along with an amazing postdoc from Yifan Cheng's lab, Hao Wu. They teamed up, and they now have structures showing that NOAD uses a part of its subunits to sense the length of flanking DNA in a very unusual way. Instead of acting as a ruler to sense it, when the DNA is not long enough, they inhibit the ATPase activity. So again, it is telling us, going back to the original question you asked about SNF2H versus BRG1, all of these remodeling complexes seem to be doing, depending on how you ask the question, either the same thing to nucleosomes or different things, right? If you ask, is the output different? The answer is yes. If you ask, do they get to the different output by doing something fundamentally different? The answer is no. Okay? And so with NOAD, we are learning that it has found yet another way to couple the basic ATPase activity of its ATPase subunit to have a different range of outputs. There is one more aspect of NOAD that you most probably have in your list of papers that you're looking through, is its preference for hexosomes. That was one of these unusual findings again because of a graduate student. Okay. Miriam Godet, she noticed on when we normally run these chromatin remodeling reactions, we have a nucleosome on a native gel, and there's usually a little bit of a hexosome, because when you make a nucleosome prep, you often have some amount of it, a very small amount of it, not fully assembled. And we usually ignore it. Everybody before her would ignore it and say, hey, that's constant. Let's not pay attention. Let's pay attention to the major nucleosome band. Right? She didn't ignore it. She noticed that NOAD, when she added NOAD to the reaction, the bottom band, which was a hexosome, actually seemed to move. Okay, so this is why it really depends on who is looking at the data. Multiple people had looked at the same data and not noticed that little band moving up, right? Or not maybe thought it was a big deal. She did. And so that got her and her collaborator, amazing postdoc, Laura Shea, to reconstitute hexosomes, clean hexosomes. And that's when we learned that NOAD moves hexosomes much faster than nucleosomes. And that it has a significance, presumably in transcription, elongation, and maybe in replication as well.
So what is missing from the octamer when it's a hexosome?
It's missing one H2H to be dimer.
And who is removing that?
That's a great question. So there's multiple models in the field, right, as to who's removing it. During transcription, Pol2 itself can remove it when it goes through a nucleosome. And Vasily Studitsky has beautifully shown this using biochemistry. So Pol2's action through a nucleosome can remove the dimer, and it can often be directional. It can remove the dimer in the direction it's moving under certain situations. The other suspect for removing a dimer are histone chaperones. Because like NAP1 or even FACT, they can loosen the interactions and pick up a H2H2B dimer. Those are the kind of the most obvious players you could imagine would remove a dimer and make hexasomes. But these hexasomes have been detected by folks like Heinekoff during transcription, right? So we know they exist. And the question has been, who moves the hexasomes? We know all these chromatin remodelers can move nucleosomes. Who moves the hexasomes? And our findings in O80 suggest that O80 might be one of the key players that actually preferentially slides hexasomes.
But that would then be after transcription and not to facilitate transcription, right? If Pol2 removes the H2AB dimer.
Yeah. So there's a lot of kind of disruption when Pol2 goes through a gene, right? And I should specify everything I'm talking about is in the context of cerevisiae. Right? There's going to be similar things happening in a mammalian context, but as you might imagine, it's going to be more complicated. But in the cerevisiae context, people have shown that when Pol2 moves to a nucleosome, it changes the location of the nucleosome. It also changes the location of the hexasome. And so what you're doing is you're opening up potential cryptic transcription sites. And so part of what is assumed to be the role of remodeler is to reset the chromatin landscape and cover up the pieces of DNA that were opened up. And so in that context, our hypothesis is that O80 resets the hexasome landscape, makes sure the hexasomes go back and cover up what they were covering. And then maybe a histone chaperone comes in and adds the dimer. Right? That's one explanation.
So to go to the finish line of this interview, imagine you're due to submitting a grant proposal tomorrow. What would you have written into that?
I'm in the US right now, and we're talking about submitting grant proposals.
Yeah.
So it's an unusual time. Yes. So let's assume I can normally submit a grant proposal. One of the areas that we are really getting into, and we have a story in bioRxiv, amazing graduate student Camille Moore has led this work, that we are getting interested in how do chromatin remodeling machines and other chromatin regulators act in a crowded context? So when we stumbled into phase separation because of Adam's work, and then because of Serena Slothy's work, we saw that you could compact chromatin. It kind of made me a little worried, right? In a good way. We're doing all this biochemistry in dilute conditions. As most enzymologists, most biochemists would look at mechanism in purified context in a dilute situation. We get really good data. We can get good kinetics. We can use the kinetics to figure out mechanism or make mechanistic models. But in the nucleus, chromatin is super crowded. Okay? The concentration of nucleosomes are estimated to be 100 micromolar. Okay? Super high, right? In a test tube, most people, our lab included, would do reactions at nanomolar concentrations of nucleosomes. So what changes when you do biochemistry in a crowded chromatin context? Or in other words, what are we missing by only doing biochemistry in a dilute situation? And so that's what the grant would focus on. It would bring together all our understanding about phase separation, all our understanding about deep mechanism, and say, hey, let's start doing biochemistry in a situation where chromatin mimics the crowdedness of what happens in the cell. And we can do that because you can make chromatin condensates in a test tube. A work from Mark Rosen's lab and our lab has shown that you can make long arrays of chromatin, condense them into droplets, and the concentration of nucleosomes in those droplets is very similar to what you find in a cell. It's 100 micromolar, on the order of 100 micromolar. So that's where the lab would go. It's going already. So that's what I would write the grant proposal on.
Okay.
To say, let's start doing biochemistry in a crowded context and figure out what's been hidden so far.
So for the last 35 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
I guess one of the discoveries that we made in the lab that is, again, it's all coupled to what we are learning about the machines, is how plastic a nucleosome is. And this was a discovery made by a postdoc, Kalyan Sinha, when he was in the lab, where if you think of a nucleosome, the traditional view of a nucleosome has been it's a packaging unit, static Lego block-like model for histones sticking with each other to form an octamer. And what he found using NMR on nucleosomes, and again, this was a super challenging project because doing NMR on nucleosomes is not a trivial task. But my colleague, John Gross, who's an NMR expert, kind of told us we should try this. And so he and I are, co-authors on this paper. And through that study, what Kalyan found, with a lot of really excellent help from John Gross, was that nucleosomes are very dynamic. And when you put a remodeler like SNF2H on nucleosomes, it makes the core of the histone octamer, the one that is buried from the solvent, dynamic. So remodelers are allosterically deforming the histone core. And if you prevent this allosteric deformation by putting disulfide bonds, you inhibit remodeling. And so there's a lot of interesting dynamics, even at the level of a nucleosome, that we are missing and that might be regulated through PTMs, to small molecules, and so on. And others have seen this now too. So it's exciting time to be doing biophysics and biochemistry at Chromatin.
So to finish, and this is my last question, what is your personal definition of epigenetics?
I'm not sure I can give you a satisfying definition.
Nobody has it.
So, if you go with the classical definition of epigenetics, is inheritance of a trait or a phenotype without inheriting or without changing a phenotype without changing the DNA sequence, right? And I would say classic development is epigenetic. When cells differentiate into different cell types without changing the DNA sequence, it's epigenetic. So to me, I would say anybody who's talking about epigenetics, define the context in which you're talking about it. Histone marks are a shorthand for epigenetics sometimes, because histone marks can modulate the genetic information in a way without altering the DNA sequence. So I can see why certain histone marks are considered epigenetic, because they can be inherited. But I would stick to the classical definition of epigenetics and then say, what are the underlying mechanisms, right? I would not go to the mechanism right away. I would go to what is the phenomena of epigenetics and what are all the different ways you can get epigenetic phenomena. Pre-onset epigenetic, right? That would be my way of looking at it. I'm more interested in what are the biochemical mechanisms that allow you to make a cell choose between different cell fates or maintain its cell fate. I told you it is not going to be satisfying.
That's true. But interesting nevertheless. So thank you, Geeta, for your time and for being on the show.
Thank you for inviting me.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening and stay tuned.